In 1865, a monk in a garden growing peas made a world-changing discovery that, at the time, was ignored. Then, in 1979, a woman leads a research expedition to a small fishing community in Venezuela, hunting down a gene that has been haunting her family for generations. Then, in 1987, Japanese scientists found something so strange in the DNA of bacteria that it cracked open a new field in genetics.
Okay, so I kind of have an idea of what this one is about.
Okay, interesting. This is kind of a first.
Rude. Okay, but
Tell me, what do you know?
Well,
what do you think?
in the history of science, there's just the one pretty famous guy who farmed peas. So if you're talking about the guy that I'm thinking of,
Well, could be anybody. There's a lot of pea farmers in history.
I'm guessing this has something to do with genetics.
Yeah, but whatever. What doesn't have to do with genetics? You know, that's all life basically. So.
Sure, but I mean, at least I've narrowed it down to
Could be anything.
all life. We're not doing non-living things.
I guess you're right about that. We're not doing an episode on rocks, at least not today.
I would love to do an episode on rocks, by the way.
Well, just say the word. I've got inflection points for days on rocks. But yeah, you're right. I mean, in general, you're also right, because today we are talking genetics, but specifically gene editing.
This is Inflection Point.
Spanning a century of reporting from C&EN, this podcast traces discoveries from our past
To how they shape our present.
and will change our future.
I'm Gina Vitale.
And I am David Anderson. Okay, Gina. So I kind of know vaguely what genes are. Like, we get them from our parents and we pass them on, our DNA, etc.
Right. Fundamentally, a gene is the basic unit of inheritance.
Okay. But I don't remember exactly what a gene is made of and how it works. I mean, I know it has something to do with DNA and chromosomes and
Yeah.
maybe dinosaurs.
Dinosaurs.
Well, actually, come to think of it, all I know about genes is basically from Jurassic Park.
Gotcha. Jurassic Park, as movies go, a great primer for talking about genetics. But let's start by talking about cells. So in your body, you have cells. And in the nuclei of those cells are chromosomes. Typically, people have 46 chromosomes or 23 pairs.
Okay, cool. And those are the kooky things that kind of look like a long stretched out X?
Yes. So what you're picturing is a pair of chromosomes. Now, each one of those chromosomes in those pairs consists of a really, really long strand of DNA, along with, some proteins. The DNA is packed in really, really tight.
Okay, got it.
Now, imagine unraveling that chromosome and looking at that long strand of DNA. Genes are basically segments of that strand of DNA. They're little chunks of it. And one chromosome can have hundreds or thousands of genes on that strand.
Okay, and we get all this from our folks?
Right, so in each of our chromosome pairs, we have one from mom and one from dad. And actually, both chromosomes in a pair contain versions of the same genes.
Okay. Sorry, can you back up? You lost me at that part.
Yeah, that's fair. So let's make this a little bit easier with an example.
Great.
Take earwax, for instance.
Perfect example. Earwax, all right. Controlled by genetics, I take it?
Well, yes, particularly one gene controls whether you have wet earwax or dry earwax.
I did not know that there were multiple types of earwax.
Hey, you learn something new every day.
New and gross.
Yeah, well, so let's pretend we're looking at a chromosome pair. The chromosome from mom and the chromosome from dad both have a gene that codes for a type of earwax, except they may not necessarily be the same version of the earwax gene.
Okay.
The one from dad could be the gene for wet earwax. The one from mom could be the gene for dry earwax. These different versions of genes are called alleles.
Okay. In your example, you got the dry earwax from mom, wet earwax from dad.
Yep.
So which do you end up with? You have one ear that's wet, you have one ear that's dry?
That would be interesting. So in that case, you would end up with wet earwax. The wet earwax allele is the dominant one. So alleles can be either dominant or recessive. If you have a dominant allele, that's the trait that's going to get expressed. A recessive allele will typically only win out if both alleles from both parents are a recessive version. Now, it's biology, so there are some exceptions to that rule, but
Got it.
we don't need to get into all those.
Okay, so if both parents had a recessive allele, meaning they both had dry earwax, you would have it too.
Yeah, and I wish I did. Having dry earwax is associated with having less body odor, which, would be cool, especially with these summers we've been having.
Okay, so are most of our traits controlled by a single gene like that? Like, is there a single gene that decides our eye color?
Actually, we now know that multiple genes are responsible for eye color. In fact, a lot of traits, at least in humans, are controlled by multiple genes. Those kinds of traits can be called complex traits.
Okay.
Now, of course, there are also traits like earwax that are controlled by one gene. These are sometimes called Mendelian traits.
Huh, Mendelian. That sounds so familiar. Huh. Well, anyway, on to my P inflection point.
Oh, okay. Well, we did a P1 last episode, David. We're doing no more urine, no more bodily fluids.
Gina, what? Hold the phones. Just hold your horses here.
I am halfway out the door, David.
I am talking PEA. P like the legume.
The legume. Okay. Oh, right. This is your mysterious pea farmer inflection point. Gee, I wonder what that could be about.
Okay, well, a little suspicious of this attitude, but anyway, let's go back to...
Is it 1865? Is that where we're going?
Yeah, let's go back to 1865.
Wow, 1800s, huh? And this has something to do with peas and genetics?
Yeah. Anything you'd like to add?
Well, no, I mean, I'm just curious. Does our story take place in, what we now call the Czech Republic?
Well, back then, it was part of what was called the Austrian Empire.
Right. Well, I assume there was this monk named Gregor Mendel.
Mendel, yeah.
Just, stop me at any time here if I'm wrong.
Yeah,
but,
all right. I will.
He's actually the namesake for Mendelian traits, which I mentioned earlier.
Okay. That does make a lot of sense.
He was curious about heredity, so he spent 8 years crossbreeding pea plants.
Pea plants?
PEA.
Yeah, thank you.
Yes. This was easier than studying people or animals since he could really precisely control their reproduction.
Yep. But did you know he
pollinated the plants himself with
All right.
a paintbrush?
Well, lucky guess. Yeah, he tracked traits like flower color and seed texture across generations using thousands of plants.
And he discovered that traits are inherited in predictable patterns. It's not just random. The plant ancestors pass down traits to the next generation. We learned all this in school.
Right. Yeah. But it's also kind of cool, right? So anyway, he publishes his findings in 1866.
And then he sets the scientific world alight with his incredible genetics discovery.
No.
What? Wait, what is it?
Genius Gina. No. I don't think so.
I'm a fan of that nickname, by the way.
This is surprising me. I thought we were kind of on the same page, or at least maybe you were reading ahead.
Well, yeah. I mean, Mendel discovering genetics. I mean, David, everybody knows this story. It changes the world.
Well, yeah, but that's the thing. Remember at the beginning of the episode, I said that he was ignored?
Guess you did say that.
Remember that right at the beginning? Of course, I'm sure you pay attention to all those
Sometimes I
little
tune out.
inflection points that I read off at the beginning of the episode.
It just takes so long. And
When he published his paper, nothing happened. The scientific world ignored his research for decades.
What?
He died thinking that maybe no one even cared about his work.
Wait, I mean, that was like a field-changing discovery. And nobody cared
that
about...
we know now. I mean, yeah, looking back, it's easy to say, but only in the early 1900s, scientists rediscovered his papers and realized that Mendel had, yeah, you're right, discovered genetics 35 years earlier.
Wow. Okay, David, I admit it. I thought I knew all about Mendel and his peas, but I did not know that. So he never knew how important his experiments really were.
Right? I mean, it's actually quite tragic.
Yeah.
Without knowing what genes are, we wouldn't be able to do gene editing.
Yeah, I mean, well, that's true. But there are a lot of steps in between Mendel's peas and gene editing.
Oh, I can't possibly imagine what they would be. Found the peas, gene editing.
Sure.
But sure. Yeah, I'll entertain this notion. Like what? Steps like what?
Okay, well, for starters, we had to figure out which genes correspond to which traits, right? For instance, after Mendel's experiments, we knew that genes are probably what give us our eye color, but we didn't know exactly where those genes were in our set of chromosomes or how many of them might play a role in expressing that trait. We also had to develop our understanding of genetic mutations.
Okay, that's interesting. Yeah, how how do mutations happen?
Okay, well, a few ways. So for instance, radiation can change the structure of DNA, but mutations aren't always caused by external factors. Sometimes when DNA copies itself, it can make a mistake.
But I thought that we had enzymes or something that could help fix that.
We do, but sometimes they don't fix all the errors. And depending on which cells these mutations occur in, like if they are in cells that form sperm or eggs, they can be passed on to future generations.
Okay, I think I get how genetic mutations can happen and that they can be passed on, but how do they in turn kind of make people sick?
Well, one common function of genes is to code for certain proteins. Basically, those genes provide blueprints for proteins that your body needs to function. If one of those genes is mutated, your body might not make the protein the way it normally would. For instance, it could make a version of the protein that doesn't carry out its intended function, or it could make a protein that's harmful. Now, a lot of mutations have no effect, and some can even be good, but we're concerned about the bad ones here.
Got it. So in order to start figuring out how to fix genetic mutations, we have to figure out which changes to which genes cause which effects.
Right. And it helps to figure out where individual genes are located. We can theorize that there's a gene somewhere that's malfunctioning, but we can get a lot more information about it once we actually find it on the chromosome.
Okay, wow. But that sounds like a lot of work.
Right. It's not a simple task. And actually, it wasn't until the 90s that we identified the exact location of any particular gene on a human chromosome.
Okay, I'm just checking my notes here, Gina, and my next inflection point actually takes place in 1979. So what's going on here?
Oh, well, David,
You said the 90s.
Your inflection points are inscrutable, so I don't know what that has to do with what I was saying.
I guess we'll just have to go back to 1979 and figure this whole darn thing out.
David. Why are we back on a boat?
This is...
we were on a boat in the last episode, and I didn't care for that.
Yeah.
I was seasick for a
You took a
day
Dramamine
afterwards.
and then you were fine.
Was I fine?
Oh, I guess sounds like you weren't. Well, this is Lake Maracaibo, okay?
Does not answer my question.
Well, in 1979, a woman named Nancy Wexler led a research expedition to Lake Maracaibo here in Venezuela.
And she was on a boat as part of this research expedition? That's why we are now on a boat?
Well, I thought it would be nice. It's a nice way to see any kind of place by boat.
Well,
go by boat, they say.
I will say we are on a lake this time instead of the deep sea, so my stomach is feeling a little bit better at least.
Some would say it's an estuary, actually. The definition of this place has been kind of fluid.
What are we doing
here,
David?
Sorry. Anyway, she was here to visit a very, very large extended family believed to have the highest concentration of Huntington's disease in the world.
Oh, okay, interesting. Huntington's is a pretty serious disease.
Yeah.
It causes nerve cells in the brain to decay, which in turn leads to trouble moving and even thinking. It's pretty rare.
Yeah, it is. But it's also genetic.
Right. It follows an autosomal dominant inheritance pattern. Basically, someone only needs one copy of the gene to develop the disorder. If one parent has it and the other doesn't, it's still typically a 50-50 chance. The problem is these symptoms often show up pretty late, like in midlife.
Right. Until relatively recently, there was no way of knowing if you had the disease until its symptoms would appear.
Exactly. So what are Wexler and her team doing here in Venezuela?
Well, she had a pretty good reason to want to learn more about Huntington's. It's actually in her own family history. So her grandfather had Huntington's, and when Nancy was in college, her mother started to develop symptoms.
Wow, that is really heavy.
Yeah. Interestingly enough, something else that runs in her family is an aptitude for science. Her mother was a geneticist who taught biology, and her dad was a psychoanalyst and clinical psychologist who founded the Hereditary Disease Foundation, which Nancy would eventually lead.
Wow, okay, so let me get this straight. This woman, who has basically a 50-50 chance of getting Huntington's, hears about this community in Venezuela with a disproportionate amount of people with Huntington's, and she goes there to accomplish what exactly?
Basically, she wants to hunt down the gene responsible for the disease. Her and her team documented over 18,000 individuals and collected 4,000 DNA samples.
Wow. I mean, that's amazing. But sorry, you said she worked with one family? A family of 18,000 people?
Right. One giant extended family. This stretches back 10 generations. They were all descendants from one woman, Maria Concepcion Soto, who lived there in the early 18th century.
Still, though, that's 18,000. That's such a big family.
Families used to be pretty big, so you imagine that someone had, I mean, this isn't necessarily a realistic example, but for the sake of the math, let's imagine somebody has 8 kids. And then each of those kids grow up, they have 8 more, and then that next generation, they have 8 of their own. Now that's already 512 people produced by those 3 generations.
Yeah, I guess that does add up.
So anyway, Nancy Wexler and her team would return to Venezuela over and over again. In 1983, because of her work, scientists were able to determine that the gene that causes Huntington's is located on chromosome 4. It was the first time anyone had mapped a disease-causing gene to a human chromosome. 10 years after that, they identified the exact gene on chromosome 4 that, when mutated, causes Huntington's disease. That gene is now called HTT, or Huntington.
So she sees this hereditary disease devastating her family and basically learns everything about it to the point of advancing an entire field of study.
Right?
That's really cool. So what happened to her? Did she get checked for the gene that she helped identify?
She didn't. She said she'd rather just not know, that living with the knowledge that she was certain she would have the disease would be too difficult.
Wow, that's really sobering.
Eventually, she did start showing symptoms and announced to the world that she had Huntington's. She told the New York Times in 2020, quote, "Having a fatal disease is scary and I don't want to trivialize that. But if I can say I'm not stopping my life, I'm going to work, we're still trying to find a cure, that would help. If I can do anything to take the onus off having this thing, I want to do it."
Wow, that's a really amazing attitude to have. And I guess we should say at the time of this recording, a company called Unicure just released positive results from a clinical trial testing a new gene therapy for Huntington's. In the trial, the gene therapy was actually able to slow the disease progression.
Wow.
If the therapy pans out, it could be the first ever treatment that can alter the course of the disease.
That's pretty incredible. I mean, that's some really positive news.
Yeah, no, it's really hopeful.
So in a lot of genetic diseases like Huntington's, we've now figured out where the problem genes are. We even know the DNA sequences of a lot of genes. How do we go about editing them?
That is an excellent question. So one of the big ways we can go about this is by making something that can bind to the problem DNA and then cut it.
Cut it? Mm-hmm. They must use a really tiny knife. Does it not damage the DNA?
Actually, once we cut the DNA, the body's natural repair processes kick in. We basically exploit those processes to fix the problem genes. And there are a few ways that we do this. One way is when we insert into the body the thing that will bind to and cut the DNA. We'll get to that thing in just a minute. We also insert a template DNA strand. So when the broken DNA repairs itself, it uses our template as a reference, rather than just building itself the way it was before.
Interesting. So we're kind of tricking our own DNA into fixing itself.
Yeah, it's a little sneaky.
Okay, so what are we using to bind to these problem areas and make these cuts in the DNA?
Good question. We have a few tools that can do this. In the 1990s and then in the late aughts, scientists developed a couple of kinds of proteins that could bind to specific DNA sequences and make cuts. But it wasn't until the 2010s that we made a gene editing discovery that really blew the doors off the place.
Wow.
This is a little tool called CRISPR-Cas9.
Ta-da!
Unlike the previous methods, the CRISPR system does not exclusively rely on proteins to bind to DNA sequences. Instead, it relies on an RNA sequence that matches up with the DNA sequence of interest. It's a lot easier to make these RNA sequences that bind to the DNA than it was to engineer those proteins.
You know, I actually know a little bit about CRISPR.
Oh, you do?
Yeah, I do.
That's exciting.
For instance, Gina, do you know what CRISPR stands for?
Yeah, it stands for complete revisions.
Interesting.
The I, what is the I?
How about this?
Individual, yeah.
We can learn all about CRISPR in 1987.
David, I actually love the 80s. I am thrilled to be in 1987.
Here we are.
This is nice. I've got my leg warmers on.
Let's turn on the TV, watch a little Miami Vice.
Love Miami Vice.
Start our electro band.
Sure.
You still have that keytar.
I have it
right?
somewhere, yeah. I've got it around here.
I'll dig up my synth. We'll have a band that will consist of just a keytar and a synth. And in Japan, scientists have discovered a weird repeating pattern in the DNA of E. coli. They stumbled upon this while they were looking for something else, so they basically kind of acknowledge it and note their findings in their study, move on. And in the years that followed, this strange thing that they noticed again and again kind of kept popping up in other bacteria, this weird repeating pattern in DNA that didn't look like anything else.
Okay, what did it look like?
In the DNA, they found 5 perfectly uniform bits all in a row, but they were separated by these 5 other totally different parts that didn't seem to correspond to anything at all.
Huh, okay, so identical piece followed by weird bit, then an identical piece, weird bit number 2, identical piece, and so on?
So on and so on, yeah.
Okay.
That's where this whole thing kind of gets its name. Clustered, regularly interspaced, short, palindromic repeats, aka CRISPR.
Wow, that is a mouthful.
That's why they needed the cool acronym.
The acronym is pretty cool.
But it wasn't until the early 2000s that someone figured out that the weird, unique pieces were actually bits of virus DNA.
But this is in bacteria, right? Surely you must mean bacterial DNA.
Not this time. No, they found virus DNA in there.
Virus DNA in the bacteria?
Yeah.
Why?
Well, scientists think it's a defense mechanism. CRISPR allows bacteria to remember a virus by storing its DNA. So now it can make a template that it can use to recognize virus DNA to know if it's being attacked by a virus.
Interesting.
Another important component here is Cas9, or
Right.
CRISPR-associated protein 9, which is an enzyme. But you can think of it like a tiny pair of scissors that latches onto the template.
Tiny knife.
There we go, exactly. So
You were onto something.
I was onto something. If virus DNA is present, the template will recognize it, and the Cas9 enzyme, or the tiny knife, will chop it up.
Okay, so CRISPR is a system that helps bacteria fight viruses? How does that help us humans?
Well, that's where 2 scientists named Jennifer Doudna and Emmanuel Charpentier come along. They say, well, what if we could put together this Cas9 enzyme with a template matching some DNA we want to edit? Give the enzyme a template and say, hey, happy hunting, little fella. That way, the CRISPR-Cas9 complex could find a really specific DNA sequence and make a cut, allowing us to try and repair it.
Wow, that is a really clever idea.
It was. That tool they made has been used in countless ways, including editing DNA that causes disease in humans. The Nobel Committee thought it was pretty clever too. Doudna and Charpentier were given the Nobel Prize in Chemistry in 2020 for their work on CRISPR.
Wow, pretty cool.
Yeah, and just like that, we have a tool that can treat every genetic disease entirely risk-free.
Okay. Well, no.
Just like that. It's that easy.
Yeah, I think
Like they say, happy hunting, little fella, and he's off.
as we have learned or should have learned by now on this podcast, it is not usually
All my problems
that
are solved.
easy. The CRISPR-Cas9 system was a huge breakthrough, and it is already helping people with several genetic diseases. But like almost every technology we talk about on this show, CRISPR and gene editing, on the whole, is not a perfect technology. There are still some risks and drawbacks.
Okay, well, when are we going to get a perfect technology?
You know, maybe one day. Maybe season 100.
Okay, well, seems pretty far away, but that's fine. So why isn't gene editing perfect then?
So it's not always safe, right? I can say that flat out.
That's Max Barnhart.
I'm a life sciences reporter at Chemical and Engineering News who covers breaking news in all things genetics, genomics, and biology.
Okay, great. So we've got a genetics expert on the call. I've got a question for him that I've always been curious about.
Okay.
Max,
Sure. David, why don't we hold your question for a little bit?
Sorry?
Because I can tell that it's probably going to be ridiculous.
Well,
before that,
it's quite important.
I want to get a clearer picture of the risks of gene editing.
There are potential off-target effects that can happen with the gene editing, and patients can have pretty negative immune responses. The vast majority of these therapies are in clinical development, meaning they have not been completely vetted for safety and efficacy. They offer the hope of a one-time permanent cure for a disease that would otherwise affect someone for a lifetime. Ultimately, many people choose to take on that risk and participate in these trials. And so long as the individual has the capacity to make that decision for themselves, who are we or anyone else really to tell them that they shouldn't do that because there might be a risk that something goes wrong, right?
And there are 2 approved drugs now in the US that use some form of gene therapy, one of which uses CRISPR, gene editing. And the CRISPR one actually treats sickle cell disease. So people with a serious condition are starting to be helped by this technology.
I mean, that's really cool, but I'm guessing this isn't like a one-size-fits-all kind of thing, right?
Right. Some people have really unique disease-causing mutations that make developing a super personalized CRISPR treatment just not viable at this time commercially. CRISPR might not be the best strategy to address, right, that specific genetic disease, but with new technologies, we're kind of getting our way around a lot of these limitations.
All right, okay, sweet. I love new technologies.
Yeah, who doesn't love new technologies?
Do the new ones also have kind of impossible to remember acronyms as well?
Actually, for the most part, no. 2 of the big up-and-coming technologies in the gene editing space have pretty simple names.
It's a relief.
Base editing and prime editing.
Base editing can make precise single nucleotide changes to DNA without breaking the DNA backbone. So it doesn't create a double-stranded break like CRISPR does. But base editing is limited in that it can only make those precise single nucleotide changes. And sometimes that's enough to fix a genetic mutation.
Okay, so that's base editing. And the other one was prime editing,
which can make slightly more complex insertions, deletions, and substitutions without breaking the DNA backbone. So that allows for even more capability and even more efficiency compared to CRISPR.
And what are we doing with these 2 new technologies?
Base editing and prime editing have successfully been used in humans in clinical trials for different diseases. And I would expect that sometime fairly soon, we will probably see FDA-approved base editing and prime editing therapies. Like, before 2030 isn't out of reach.
2030, that's, I don't know, in like 5 years.
Exciting, right? To be clear, that is just a guess. We don't know when it'll happen, but it's exciting that the use of such groundbreaking new tools is actually in sight.
Yeah.
And further down the line, we may see even cooler ones. Max is always covering those. What's on the docket for the future, Max?
One of the technologies that I covered last year is bridge editing, which is like one of the newest in this space of gene editing technologies. And with bridge editing, it's possible to make really big DNA changes, like large insertions, large deletions, even rearrangements to the sequence or like the order of large components of DNA. And that could be really useful for one day treating diseases like Huntington's or like ALS, amyotrophic lateral sclerosis, Lou Gehrig's disease. These are diseases caused by the expansion of repetitive sequences in DNA. And with bridge RNA, you could like shrink those repetitive sequences and get rid of them and have a permanent cure. Now, something like that is a ways off.
Okay, all right. In the future, we may be able to permanently cure a bunch of devastating diseases. I guess that's cool or whatever.
Cool. It's more than cool.
Yeah, it's kind of cool.
I mean, I would probably use earth-shattering.
Nifty,
probably
maybe?
the word that I would use.
Earth-shattering, yeah.
Yeah.
Well, sure. That's great.
Sure.
But I would like to circle back to my question from earlier that was so rudely ignored. I would like to know what I would look like with brown eyes. Can we make that happen?
Oh, brother.
Maybe one day you'll be able to slightly change the colors of your eyes, or like, you know, for folks who suffer from like male pattern baldness, you know, maybe you could get a gene editing cure for that, perhaps, right? I think it's feasible at some point that you could do gene editing to like change how you look. That's not soon. I think there's going to be a lot of conversation about like if that's allowed or not. And generally speaking, I don't think it should be, because it's not trivial to do this stuff. It is, as I've said, dangerous to gene edit yourself.
All right, well, whatever. I guess I'll just go on Amazon and order some colored contact lenses.
I think your eyes are nice the way that they are, David.
Oh, thank you, Gina.
And as we've seen through this conversation with Max, this is a pretty amazing time to be following the gene editing space. Unprecedented discoveries are being made right now. There's a lot to be excited about.
I think it is absolutely fair to say that we have entered the era of genetic medicine, right? One of the great parts of it too is that some of the most like vulnerable of our population is going to be the folks who get the benefits of gene editing therapies first. And I think that's a beautiful thing.
That is so amazing. Thank you, Max, for injecting a little bit of hope into our podcast.
No kidding. Yeah, it feels so good to be delivering some good news for a change.
Yeah.
I feel like I'm glowing.
Me too.
It's not just the compliment about my eyes.
Oh. It's nice to feel hopeful about the future. We should cling to that feeling as long as we can.
Well, on that wholesome note, Gina, are you ready to hear the inflection points for the next episode?
I'm ready.
So here we go. In 1928, there's this professor who goes on vacation and returns to his lab to find something he later calls mold juice.
Mold juice. Okay.
Yeah.
It's kind of disgusting.
30,000 years ago, there was some DNA literally frozen in time that held on to a secret that scientists only now recently uncovered. And in the 1960s, researchers found an organism thriving in one of the harshest environments on Earth.
Okay, interesting. Mold juice.
Yep.
Don't love the sound of that.
It pays off.
30,000. You said something was literally frozen in time. Are we gonna go somewhere?
Frozen in time, 30,000.
Are you gonna
years
drag
ago.
me somewhere really cold? Because I really, I don't.
Could be very
I'm much more of a
cold, yeah.
tropical kind of girl. I don't know that
hey,
I'm.
you might love one of the harshest environments on Earth. We're also going to visit that next episode. I will say, it's going to get very extreme.
Good.
So yes.
Okay, so this is like I should just kind of pack for all.
You know what? Just follow your instinct.
I should just really. Great. Okay. Well, I will.
You'll need some jacket. You'll need
Got it.
some, I don't know,
Okay.
fireproof
I will
suits, something.
Pack the biggest bag I can find.
It's going to be an adventure.
Inflection Point is a podcast project from Chemical and Engineering News.
Chemical and Engineering News is the official news outlet of the American Chemical Society.
Music by Kirk Onstad and Shutterstock.
Written, produced, and hosted by Gina Vitale and David Anderson.
Additional audio production by Jeremy Barr.
Thanks for listening.